maybe melts the DNA helix a little, binds with the DNA, binds with the histone or something. Those are the transactivation regions. And remember, this guy here shows you that these guys have a DNA binding region. They have a region that can bind to hormones or metals or whatever that affects their binding here. But then they also have these transactivation regions which activate the DNA near the enhancer site, near the binding site. Make sense? So transcription factors have three important parts, right? The dimerizing region, the DNA binding region, and the transactivation region. Yes?
You said it was important if there were two basic regions. Is that just for specificity?
The two basic regions, of course, the longer the basic regions, the bigger the specificity. But the other reason for two is if you have one, it just forms a helix and it's kind of straight. And the DNA is round. The DNA helix is circular. So if you have these two with a break, those two alanines, it allows those two basic regions to bend. So they can actually, instead of going like this on the DNA, they actually bend right around in the groove. And when they do that, then that can extend the specificity binding, the recognition site, which extends specificity. Okay. Anybody have a question about DNA binding? There are others, but those are the basic forms. And you can find, as I said, zinc fingers, there's certainly hundreds of transcription factors in eukaryotic cells that have zinc fingers. So the motif that's been used over and over again to bind DNA. Okay. So we're going to start talking about a little bit more about transcription. We've said that we've talked about DNA replication, we've talked about transcription. I think we've talked about replication of this recognition of the promoter region a little bit by these DNA binding proteins. And when they do, they begin to transcribe that gene. And what we're going to do now for the next day is we're going to talk a little bit about this primary transcript and what it looks like, and about what happens to that primary transcript before it gets out into the cytosol to get translated. Don't forget, transcription is DNA to RNA, translation is RNA to protein. Don't miss them up, because I'll use those words in a quiz, and if you don't know them, you'll miss them. Okay, so transcription of the RNA. We said RNA polymerase is recognized as a promoter. Here's RNA polymerase that's there. RNA polymerase has this tail, the CTD from our G-terminal domain, with all of those repeats. In order to begin elongation after initiation that promoter, it has to be phosphorylated. There's those 100 base serines and threonines. Many of them are phosphorylated over and over again. And when they are phosphorylated, they lose their affinity for all of the transcription factors for the DNA at the promoter region, but they gain new affinity for other proteins. And there are several proteins that get on there. Capping factors, splicing factors, and cleavage factors, and we'll talk about all of those as we go. So, as RNA transcription or elongation begins, the very first thing that happens is the RNA is modified, messenger RNAs are modified. And the way that they're modified is they have a cap that's put on the 5' end of the RNA. The 5' 7-methyl G cap, 7-methyl guanosine cap. And that cap occurs on that 5' end only on polymerase II transcripts in eukaryotic cells. Why? Because only the polymerase II, the catalytic subunit of polymerase II, has this carboxy terminal tail. Polymerase I and III don't have it. Only these guys carry these proteins, these factors on there that modify, or these RNA modifying factors. So as transcription begins, there's the new RNA being made. As that nascent RNA molecule first is made, the capping factors essentially that are riding along on that tail jump off and bind on that RNA and put on that cap. Okay? This just describes that tail to you. We've talked about that. So what is this 5' cap and how is it put on? 5' cap is the addition of a base or a nucleotide to the 5' end of a growing messenger RNA. Remember, this is the 5' end of the messenger RNA. There's the nucleotide. And that 5' there it is right there, the 5' transfer. And that 5' transcript has a triphosphate on the end. Why does it have a triphosphate on the end? Go ahead.
Because it's fine when it's bound, and then I guess it gives it enough energy so we cleave it.
Yeah, that's true, but why is the 5' one? Right? We're talking about a messenger RNA, and the 5' end and the 3' end. What's on the 5' end of DNA, if you make DNA? Monophosphate. Why? Why? When you make DNA, what's on the 5' end? Primer. What's on the 5' end of the primer? A 5' nucleotide triphosphate. Because it's the first triphosphate that's put together. DNA doesn't have one, it has a monophosphate. Why? Well, one thing is made with a primer. Why is it made with a primer? So it can what? I didn't ask you that. I'll have to ask you on the next quiz. Why is it made with a primer? What does DNA polymerase always have to do? First step, proofread. It has a primer for proofreading. So when you make DNA, the first deoxynucleotide triphosphate is not on the 5' end. There is an RNA on the 5' end. It has a triphosphate. The DNA is there, and then of course when you process it, you remove that primer, right, and extend DNA. So why does this guy have a 5' triphosphate? Because RNA doesn't proofread. RNA polymerase does not proofread. It just finds a promoter which lines up polymerase, right? Just sterically puts the polymerase there on that DNA. So because of the size of the footprint of the polymerase and where the catalytic site is, it lines it up at nucleotide where that codes for amino acid 1. It lines it up with the very first triplet that goes for an amino acid, right? Okay. And it just starts. Then the first one's a nucleotide triphosphate. So there's that guy. The enzyme that actually caps is the capping enzymes. There's actually 3 enzymes involved in doing capping. Here's GTP, right? And so the 5'7-methyl G-captor added right after transcription. I showed you that about 25 nucleotides. I've soaked them out, and that capping complex took on. The capping enzyme associates with the phosphorylate. I told you that. Don't have it. Cytosolic phosphate detail. Okay. So essentially what happens is here's the first enzyme. It's called guanylyl transferase. And guanylyl transferase is it transfers a GTP molecule onto the 5' end of the RNA. This shows you. This is the sugar phosphate backbone right on the RNA supposedly. So this is the 5' nucleotide. Now, when it does so, what happens is guanylyl phosphate takes this guy here, right, and it removes a pyrophosphate, so it leaves one of those phosphates. It takes this triphosphate and it removes a phosphate. You can see there's a purple phosphate and a big pyrophosphate. And then it puts together these enzymes, right, and forms a triphosphate in the middle. So essentially what you have is a guanine cap on the 5' end of the RNA, and it's put together kind of backwards. So now, this is now put on the end of the RNA molecule, what does this RNA no longer have? What does it no longer have? It no longer has a triphosphate end, right? If you look at a newly synthesized RNA molecule, tRNA, ribosomal RNA, messenger RNA, they all have this 5' phosphate on the end. What does that 5' triphosphate do? Can anybody guess? A phosphate on the 5' end, what does it do? Well, remember, we've talked about exonucleases. 3' exonuclease, 5' exonuclease. The 5' exonuclease recognizes that triphosphate and recognizes this as a nucleotide and begins to cleave in, right? So when you put this cap on here, there's no longer phosphate because you've taken this G and you've inverted it so the phosphate becomes part of the linkage between them. So these guys are linked by the triphosphate. There is no phosphate cap on the 5' 5' end of a messenger RNA. And because there's no 5' cap, or no 5' phosphate, the endonuclease doesn't recognize this. So putting this cap on stabilizes that messenger RNA from degradation by exonucleases. Make sense? So the first enzyme is this guanylyl transferase that makes this linkage here. The second enzyme is an enzyme that actually methylates the cap. So there's the 7-methyl G, this is number 7 right there. Right? This is 8, 9, this is number 7. That atom, and that nitrogen is the 7th atom in this purine ring, and there's a methyl group on there. So there's a 7-methyl G that's put on this top. So that's the second enzyme, the methylation enzyme. The third enzyme is another methylation enzyme, right? And essentially what that second enzyme does is it methylates this penultimate, or maybe a couple of the nucleotides that are after the guanine. So this would have been nucleotide number 1 and 2, right? And it methylates them. Where are they methylated? Where are they methylated? On the 2' hydroxyl. So these are called 2'O methylases. So these 2'O methylases come and methylate the ribose. Right? This guy here is actually methylating the base. This is methylating the ribose. So now messenger RNAs have this 5' cap, and the 5' cap has a 7-methyl G, cap without a free phosphate tail, and the riboses, the second and third ribose, are often methylated. If this second one is adenosine, it is methylated. If it's not adenosine, then there will be one methyl group. Most transcription, the majority of transcription in eukaryotes contains an adenosine. Okay, so we've methylated our mRNA. So the 5' cap distinguishes mRNA from other RNAs. It's now different than DNA and ribosomes. The cap, I said, has to do with the half-life of the RNA. Cap also is extremely important, and we'll see in protein synthesis. The cap binds a protein complex. The CPC, the cap binding protein that helps mRNA to be processed. That's true. It's important for processing. Nascent RNAs, nascent messenger RNAs do not leave the nucleus of a eukaryotic cell until they're processed. They have to be processed first. And the binding protein to that cap ensures that that processing happens. It's important in translation, which is what I started to say. If you take a eukaryotic messenger RNA and remove the cap, it won't be translated. Ribosomes bind and recognize the 5' cap. That's very different than in prokaryotes. We'll talk about those two different types of translations, prokaryotes and eukaryotes. But in eukaryotes, that 5' cap is needed for translation. And then this is what I told you, the 5' cap prevents it from degradation by endonucleases. So it actually has 4 functions. Right? What are the functions of the 5' cap of messenger RNA? There they are. Any question?